Good afternoon, everyone. Thank you for being here. Welcome to the 28th Annual Roth Conference. We're sure that this is going to be one of our larger ones. We have almost 4,000 people that have registered for the next 3 days. We hope that you all find it informative and educational, and maybe even find some good ideas to invest in. As Byron said this morning when the bike ride was starting, we're kind of calling today an opportunity for us to celebrate heroes. Byron talked about the Challenged Athletes Foundation and the heroes there and how they've had setbacks but have recovered from that and are now doing what they really want to do in the athletic regime. The bike ride was in the name of our personal hero, our brother Dwayne, and we thank all of you that participated in that. And I'd also like to welcome our sister-in-law, Renee, who's with us today. So thanks, Renee. So the first event that we have this afternoon is another hero of medical science. J. Craig Ventner has done a lot of things. I'm not even going to start to go through them. I'll let Michael Lerner do that in a few minutes. But I've heard it said, and I agree, that one day you're going to be able to say, I heard a Nobel laureate give a speech at the Roth conference. Craig has done a lot of things in the genomics area and a lot of things, other areas that he doesn't always speak about, but truly one of the leading scientists of our day. To introduce Craig, I have the pleasure of bringing Michael Lerner, who is a partner at Lowenstein Sandler, who is one of our lead sponsors for the conference. So following this, there will be about a 10-minute intermission, and then we'll go on with the next event, which is the Tig and Oz, talking about the events at Benghazi a few years ago. So please welcome Michael Lerner.
Thank you, Ted. As chair of the Life Sciences Group at Lowenstein Sandler, it's once again our honor and pleasure to sponsor the 28th Annual Roth Conference. And it's a great honor to me personally to be able to introduce today's keynote speaker, Dr. J. Craig Venter. As Ted said, Dr. Venter is one of the world's best-known living scientists. He's been called a leading scientist at the edge of the revolution in biology and raw data. Famous for sequencing the human genome at the turn of the millennium, Dr. Venter considers his work the starting line for the future of medicine. More recently, Dr. Venter's company, Human Longevity, Inc., is taking the power of the human genome and combining it with the next generation of DNA sequencing and stem cell advances to decode and fix the factors that contribute to aging and disease. Dr. Venter's story is a remarkable one, not just for what he has accomplished in so little time, but for the journey he has taken to where he is today. He began his formal education after a tour of duty in Vietnam from 1967 to 1968. After earning both a bachelor's degree in biochemistry and a Ph.D. in physiology and pharmacology from the University of California at San Diego, he was appointed a professor at the State University of New York at Buffalo and the Roswell Park Cancer Institute. In 1984, he moved to the National Institute of Health campus, where he developed Express Sequence Tags, or ESTs, a revolutionary new strategy for rapid gene discovery. In 1992, Dr. Venter founded the Institute for Genomic Research, a not-for-profit research institute where in 1995 he and his team decoded the genome of the first free living organism using his new whole genome shotgun technique. In 1998, Dr. Venter founded Celera Genomics to sequence the human genome using new tools and techniques he and his team had developed. 2 years later, in 2000, President Bill Clinton declared a tie in the race to map the human genome, giving credit to both Dr. Venter and his publicly funded rival, Francis Collins. In 2001, Dr. Venter's pioneering work was instrumental in helping sequence the genome of the anthrax strain mailed in the attacks that killed 5 people. His work helped bring perpetrators to justice. Currently, Dr. Venter and his team at the J. Craig Venter Institute continue to blaze new trails in genomics and have published numerous important papers in such areas as environmental genomics, the first complete diploid human genome, and his most recent groundbreaking advance, creating the first self-replacing bacteria cell constructed entirely with synthetic DNA. He has also been named by Time magazine in 2007 and 2008 as the 100 on their 100 list of most influential people in the world. He was also profiled on 60 Minutes in 2010. The scientific advancements that Dr. Venter has provided over an incredibly short period of time have laid the groundwork for discoveries that are sure to positively affect the way we live today and tomorrow. Perhaps Popular Mechanics magazine described it best when they said, "From mapping the human genome to creating the world's first man-made bacteria cells, Dr. Venter achieved a lifetime of innovations in just 10 years." With all that, without further ado, I'd like to welcome one of the leading scientists of the 21st century, Dr. Craig Venter.
Well, thank you for the very kind introduction, and Ted, thank you for inviting me to spend a day up here in this lovely place. So this must be the group that doesn't ride bikes, play tennis, or golf, or you're all recovering from it, which is equally good. So there's a lot happening in this space right now, and so I thought I'd put some of the work into context that you'll be hearing about soon about synthetic life and how that's important to understand even the basics of the human genome as well, and then what we're doing with all that at human longevity. So this building you see is our headquarters of human longevity in La Jolla. And this is the Venter Institute, which is a not-for-profit institute that's now 22 years old. It's on the UCSD campus on the corner of Torrey Pines and Torrey Pines. So if you live in La Jolla, you've seen this building, and it's the world's first carbon-neutral research building, so we're trying to practice some of what we preach in terms of the environmental aspects of this work. So when we first started reading the genetic code, we refer to it as digitizing biology. So we're converting the genetic alphabet of ACs, Gs, and Ts into ones and zeros in the computer. And ever since the first genome in '95, the computer databases have really been filling up at an ever rapidly changing pace. And one of the underlying assumptions is all those ones and zeros actually represent biology, and in an absolute sense, but it's never been proven. And so one of the early things we set out to do was to try and prove that. So when we sequenced in '95 this first genome, we actually did 2 that year. One was much smaller than the other, and we just asked some very basic fundamental questions about how many genes were required for life, and could we get a molecular definition of life. And that started us down this route, trying to work out whether DNA contained all the necessary information for cellular life. Obviously, if we're trying to interpret everything about you from your human genetic code, we need to know the answer to that question. And we set out some unique ways to do it. We decided to start with the ones and zeros in the computer and see if we could go the other way and recapitulate life simply from this digital world. We started with a small virus that kills E. coli, 5X174, and we figured that would be a good test for synthesis methods. And it actually took a while to do because the machines that write the genetic code create lots of errors when they do this. We had to develop new tools for correcting those errors. But the exciting thing came when we had this 5,000 letter piece of chemically made DNA from 4 bottles of chemicals. And we injected it into E. coli, and E. coli immediately read this chemically made piece of DNA as though it was just natural DNA, produced proteins. The proteins formed the viral particle, killing the bacteria and producing billions and billions of copies of themselves. So that allowed us to show that we could actually use synthetic DNA in life. But our goal was actually to make an entire living cell chromosome, not a small virus. So the next almost a decade or so was spent developing the techniques to go from making something 5,000 letters to this first chromosome we made was 1.1 million letters of genetic code. And that took a lot of changes, a lot of new tools. I won't go through all of those right now, but we can now, and at Synthetic Genomics, my other company, they have the ability to rapidly make entire chromosomes. So while the first one we did took us a while to do, we now make several a month. The key challenge was how to boot up a chromosome. Nuclear transplantation has been used for almost a century, where you pop a nucleus out of cell and you can move it to another one. In bacterial cells, the chromosome is just in the cytoplasm, so there's nothing to pick up and move. And so we had to develop all these techniques for doing chromosome transplantation. And we found when we did this, when we transplanted a chromosome from one species to another, it actually converted that entire cell or species into that of the new chromosome. So let me walk you through it because it's kind of fundamental, and we have this very high-tech movie to show you what happened. So when we had the complete 1.1 million base pair chromosome made from 4 bottles of chemicals, we inserted it into a recipient cell. And as soon as we did, the DNA started to be read and produced proteins. Some of the early proteins that were produced are called restriction enzymes. We had the system balanced so they would recognize the chromosome that was in the cell as foreign DNA and chew it up. So now we have the body of one species and the genetic software of the other. And as this went on, these turned into these new cells that every single molecule in the cell derived from that new chromosome that we put in. There wasn't a single molecule left from the original cell. So this is what we reported in 2010 as the first synthetic life form driven by a synthetic chromosome. And there are several very critical aspects of it. There's everything we know in terms of gene regulation, et cetera, occurs in this cell. So all that is coded for in the linear chromosome. So we think this helped prove that life is a DNA software system, and that if you change the software, you change the species. Now, this helps explain a lot of different key things in biology, and also protection mechanisms we have. For example, if you had fish for lunch, and by consuming those fish chromosomes, you started to transform your own into those of the fish, species wouldn't last very long. So bacterial cells develop restriction enzymes to protect themselves, and we have all kinds of mechanisms that protect us. Even though we consume large amounts of foreign DNA, it doesn't transform our cells. So the next stage was to try and see now, once this control experiment was done, whether we could design a new species from scratch based on the fundamentals of biology. And that's what we spent the last 5 years doing. We have computer software for trying to design this biological software. And we set out, we had a contest within the Venture Institute and Synthetic Genomics to see if we could, who could design a species that would actually live based on all the fundamentals of biology. And it turns out nobody could. Not one single design worked. And it's, you'll see it's because we don't know about a third of the fundamental elements that control life. So we had to do this iterative design build process where we would add back unknown genes until we could get to a living cell. But we finally recently achieved that, and it has the smallest genome of any living species. You will probably never find anything like this in nature. And it self-replicates and has produced several billion copies of itself. We also designed a unique 16S RNA molecule so it would be identified as a new species. But out of those limited number of genes, 149 are of unknown function. So that tells us we don't have a knowledge of the fundamentals of biology at a level for designing new species. But keep in mind, this cell is the smallest cell with the smallest genome, and we're trying to extrapolate from the human genome with over 20,000 genes and trying to understand functions at various levels. So this paper is coming out in Science in about a week and a half. It will be the first designed species from scratch. And it'll be an exciting step forward in this field because it shows that we now have a new starting point. And these are actually pictures of these cells that can rapidly make more copies of themselves. So this takes us back then to the human genome, which is infinitely more complex, even though we have a microbiome that has maybe 100 trillion or so microbes in each of us as well. And we're trying to go from that DNA sequence at the same level to try and understand life, only now much more complex life, trying to predict human traits, human diseases, life outcomes from that genetic code. So we decided we had to start at a new point. In 2007, the Venture Institute published the first complete diploid genome where it describes the chromosomes from each parent so we can understand that complexity. And we've decided to put that together in a scale that's never been attempted before because we need to understand the 3% or so variation in the whole human population to be able to understand and make these predictions. So a number of things have changed since that first genome 15 years ago. That cost us about $100 million for that first genome. It took 9 months to do. We had to build a $50 million computer to analyze the data. And those areas have changed dramatically. We are now sequencing a genome roughly every 15 minutes at Human Longevity for certainly less than $2,000 a genome. It's more like $1,500 a genome. You can buy a card for your PC that has all the compute power of our $50 million computer 15 years ago. It would distribute in cloud computing now. It makes it possible to do things we couldn't even dreamed of before. And on top of that, we've added a complex machine learning approach that I'll tell you about. So we have 3 different campuses now for human longevity: La Jolla, Singapore, and the machine learning group is in Mountain View, California. We built the world's largest human genome sequencing facility. It's now generated over 25,000 complete human genomes. We have clinical and phenotype data. And as I said, we're adding them roughly 1 per 15 minutes. People thought that genomics wasn't going to be a big data problem. We're now about 3.5 petabytes of data just for the ACs, Gs, and Ts. And when you add in all the MRI imaging and other things, all of a sudden the most jaundiced computational person starts to take this area seriously. We're in the 1% user group at Amazon Cloud, up there with video streamers and porn sites. We think we're the only legitimate program up in that area. So it's using massive amounts of computing to track this data. Now, one of the things in this field that's very confusing is people have bastardized the word genome to mean any sequence that comes from a genome versus a complete sequence of a genome. And Even the scientific journals are very imprecise now. There was recently a paper in Nature claiming it was the sequence of 1,000 human genomes. It turns out it was sequenced from 1,000 human genomes, and it's this arrow where you see the blue line here. They had barely covered 30% of those genomes, but the title implied that they had completely sequenced them. There was an announcement last week about a company said it was making the data available from 10,000 genomes. In fact, it was average data from 10,000 exomes and represented just a tiny fraction of the data. 23andMe implies it's gene chips or genomes. So there is a lot of confusion. If it's your genome being analyzed and you want accurate data, you don't want such confusion. So at HLI, we have a standard where we sequence every base pair in the genome an average of 10 times. And we're showing what clinical accuracy can be and needs to be. The whole human genome is not accurately covered. So you see these gray areas on these chromosome maps. They're mostly around the centromeres, where there's a lot of repetitive data, no genes, and not very accurate data. But in the parts of the genome that contain genes, you see all the blue bars. You can see we have very good coverage of these with a very, very low false positive and false negative levels. So, and we're sharing all this data with the FDA, and we've given them a large deposit of genomes so they can judge accuracy of other genomes. And so they're using this data as part of things. At about 8,000 genomes, we pass by the level of all common variants in the human population, and more variants in our database than in the entire public domain put together. And now we've surpassed that by about an order of magnitude. And that allows us to do some unique analyses of invariant bases in the genome, that if you have a mutation at one of these sites, you will either have a cancer or something that's incompatible with life. And so we have a totally different map of the human genome that was never able to be generated before based on this data. And for example, just with transmembrane proteins, we can track these across the genome and find sites that are invariant. So this enhances when you're looking for a variant or set of variants associated with traits or disease. We have a whole new place to look versus just trying to average information. A gene chip from Illumina 23andMe measures common variants in the population, things that everybody shares and therefore can't be the cause of key traits and disease. We now have well over 100 million variants that occur in the database one time. And what we're finding as we do this allele frequency associated with disease and key traits, they're all in the extremely rare variant population, not in what everybody's been measuring for the last 15 years of common variants. We each have a set. If we sequence your genome now, we will discover about 8,000 new extremely rare variants that describe some essential things about you versus everybody else in the population. And I can give you some examples of these. We can now do extremely rare disease discovery very quickly with just either a single patient or, in this case, a patient with the parents. It's called a trio. This is a child from Rady Children's Hospital in La Jolla. If you can read MRIs, you can see the brain is pretty severely scrambled. The skin is covered with various tumors. And it's a disease called Delleman syndrome that there's only about 30 cases of in the entire world. And so nobody had characterized it. But just with using our new tools, we found a number of combinatorial variants associated with it, several that determine the development of the brain and therefore why it was so scrambled. And then we found an NF1 mutation. NF1 is the gene associated with elephant man's disease that led to all his tumors, that in this case explains all the tumors all over the child. And it's quite interesting because we had both parents' genomes and the child, and neither parent has this mutation. So this was a spontaneous mutation in this child that combined with these rare neuronal development mutations. You can see why it might only be 30 cases in the world of something like this. It's a very rare happenstance. But also tells us that the gene sets we have are not just the ones we got from our parents, but we all get several thousand spontaneous mutations going through the developmental process. So you truly need to know your genome to understand things. You can't just depend on your parents. We've been doing the same thing now with comprehensive cancer analysis. And at HLI, we have the most comprehensive program in the world where we sequence the entire genome of the patient, the genome of the tumor at 3 times the level of the patient. We sequence the immune system. We sequence RNA in the tumor. We isolate T cells from the tumor, and we do liquid biopsies. And this gives us a very quantitative tumor burden. So in this case, it's an individual with HPV-caused head and neck cancer. This is all in collaboration with Ezra Cohen at UCSD. So we found 25,000 mutations across the tumor genome. We found about 315 that changed protein coding regions. But in the patient's genome, we found 3 mutated oncogenes and genes associated with immune suppression. And from the microbiome, we found HPV16 present. So just from his genome and the microbiome, it was more a question of when, not if, he would get cancer. And sequencing the tumor, we found where the virus had inserted 4 different places, disrupting genes. If we sequence your genome, we find these oncogene risk, and you have HPV16 present, even though you're not a young man or a young woman. You might talk to your physician about getting the HPV vaccine to see if it could prevent cancer in adults. It hasn't been tried yet, but we can take all this data on these mutations into pathway analysis and find novel sites, not just in the mutated proteins, but in the pathways. And equally, if not more important, we have a cancer vaccine program where each one of these mutated proteins, the name for them is neoantigens. It's why your immune system recognizes tumors. These proteins are different from all your normal proteins, and they're recognized if you have a good immune system as foreign DNA. Most of us get tumors all the time, and our immune system is eliminating them. If you walk around in this nice sunshine, you can get 10,000 or more mutations in your skin genome relatively quickly. Fortunately, we replace our entire skin cells every couple of weeks, so we're getting rid of those, but your immune system is always clearing those. We're taking these neoantigens and we select T cell populations. There's now 2 patients in La Jolla getting these as therapy for their resistant tumors, and it's showing a great deal of promise. So the genome will be the dictator of going to immune therapy or not, or selecting other chemical targets. As we try and get hundreds of thousands of human genomes, because the cumulative data is already showing an immense power, we decided the key thing was to add phenotype data to the analysis. So we formed the Health Nucleus as a way to not only change medical practice, but also to get a very large data set of unique phenotype data that we can use for interpreting the human genome. So we're trying to change medicine from a reactive program to a proactive program, and I'll give you several incidents of this. Instead of waiting for symptoms to occur, by detecting things early, predicting risk from the genome, we can find things at a very early stage. So in the age group of sort of 50 to 74, if you're a male, you have a 30% chance of dying during that age period. If you're a female, you have a 20% chance of dying in the US. As you can see, about a third in each is from cancer, and a third or so is from cardiovascular disease. So So just being able to predict who's at risk for cancer, who's at risk for cardiovascular disease, gives a chance for a major change in a preventative medicine paradigm. So we're integrating all this data together with the genome, the microbiome, and all kinds of different tests to make these predictions. So in the first floor of our building at HLI, we have our clinical center, the Health Nucleus. We have the latest GE 3T MRI. And now with these new differential water detection techniques, we can get photos and images like this without any contrast media. This is 8 minutes in our MRI to get an entire map of your brain blood vessels with no injections, no contrast. One relatively young woman, and now that I'm a year from 70, everything is relative to that. She was in her 50s. We found this aneurysm. You can see things like this just show up quite clearly, that obviously there was no way for her to have awareness that she had this, and she's working with vascular surgeons right now on how to deal with it. Alzheimer's disease is actually pretty easy now to detect in the MRI. The brain here on the left is a normal one. I don't know if it shows up or not. This yellow area is the hippocampus. And in the Alzheimer's disease brain on the right, you can see that same region has really shrunk and there's all this black space around it. Our colleagues at UCSD, under the leadership of Anders Dale, have developed this quantitative algorithm that takes these and other brain images. It's called Neuro Quant, and gives the exact precision volume of different brain regions and can detect changes in as little as 3 months as changes take place in the brain. And quite often, we can see things a decade or more before anybody has experienced symptoms of dementia. The same restriction spectrum imaging now allows cancer to be diagnosed directly in a non-invasive MRI image with no contrast media, nothing. This is diagnosing prostate cancer just straight from these MRI images that matches the histopathology in a very precise fashion. 20 years ago, when people started doing imaging, particularly with whole bodies, they were doing CT scans that could not detect normal tissue from cancerous tissue. And it created this whole knee-jerk response that you should not do pre-screening because people chase down a lot of things that they couldn't tell whether they were cancerous or not. Now this is unambiguous in terms of prostate cancer and some other areas that are being developed. We're seeing not an insubstantial number of various kinds of brain lesions in tumors. Some of these are just vascular in nature, like this cavernoma, but these have a tendency to bleed and cause the equivalent of strokes or other problems. This is a very interesting one. I don't know if you can see, it's a walnut-sized bright red growth right on the top of the brain. By the way, all these were found in individuals between 30 and 80 that considered themselves perfectly healthy and disease-free that came in to go through these programs. This is one of the most interesting ones, a 58-year-old male who just, the only symptoms he had was this sort of persistent cough. He got a chest x-ray each year, but it never showed anything. And we found this 5-centimeter tumor right underneath his breastbone. And you can see with the differential imaging, it lights up, like a light bulb, that it's cancerous. And so this was removed a week after its detection in the Health Nucleus using robotic surgery. And he was home a few days later, completely tumor-free. The pathology showed it had just started to penetrate the tissue, and the view was that if it had gone on another 6 months or so, it would have gotten to a stage where it would have needed extensive chemotherapy with possibly a very different outcome. 1.3 million people get diagnosed with cancer each year in the US. They didn't all instantaneously get cancers. They instantaneously had symptoms that made them aware that they had cancer. 6 months later, this guy would have started to have a different kind of symptom, or this would have started to metastasize to other areas. So when we get tumors for our comprehensive cancer program, they're usually at stage 4 when they're diagnosed. We're diagnosing tumors at stage 0, stage 1, where they can be removed, and that's the end of it. We get all kinds of quantitative data that you may or may not want, but we can very quantitatively measure visceral versus peripheral fat. And this is becoming very important. Those of you who are athletes, we can tell you exact amounts of muscle and types of muscle tissue. But with the MRI, we can tell you quantitatively how much liver fat you have. So normal is less than 4%. Their record now for the health nucleus, again, of people mostly from the San Diego area that think they're healthy is now over 38%. But we have quite a spectrum, and this is one of the leading causes of disease that without this technique, the only other way to get this data was from a liver biopsy. This changes very dynamically. Mine was below 4%, and then I had another scan after the end-of-year holiday spectrum with less exercise and more alcohol, and it went up 3%. So I've been trying to diet a little bit before I get it measured again. But it goes up and down rapidly. But the only way to really lose your organ fat is by calorie restriction. One of the leading obesity experts we're on, I think, a similar area where you guys had your ride today. It was a 30-mile ride, not a 40-mile ride. And we were halfway through the ride, and he said, you know, exercise won't get rid of that organ fat. And I said, you couldn't have told me 15 miles ago. So exercise helps, but, you know, it's really calorie reduction is the only way to reverse this. So I'm doing my own experiments now to see. We've had 2 ovarian cysts in women that were completely unaware of them. This is the smaller of the 2. The other one was the size of a football. And whether they're cancerous or not, the biggest problem with these is torsion. They can actually twist and just rip blood vessels. And women usually present with massive internal bleeding with these. So again, these are just simply detections and easy treatments. So we're feeding this kind of phenotype data in with the genome. We've had a couple cases of polycystic kidney disease where there's a clear-cut genetic association. We can find the reason for it in the genome. We can find the outcome on the MRI imaging. But we're trying to mix all this data together with machine learning to predict things in a complex fashion going forward. So I will show you a couple examples from it. And we're just starting this coming week to deliver our first whole genome reports and health nucleus reports to clients. So it's taken a while to get it right, but if you can get one from Illumina, it's 16 pages long, and 12 of those pages are boilerplate. Ours is about 250 pages long and gives you really comprehensive information about you and your genome. But from your genetic code, we can predict your height, your weight, your BMI, at least what your genome is coded for, your eye color, your skin color, and your face. So these are all things that just come straight from the ACs, Gs, and Ts. From a simple digital recording of your voice, we can readily identify what sex you are, your age, and your height. Height was a surprise to me from the voice, but all these things are interrelated to your overall volume. I've asked the team to record some things from the television show The Voice to see if they can, because there's always, I don't know if anybody watches that, there's always some rare surprises there, but you can see there's this fuzzy level in the middle that you can't make 100% distinction. We lose sex chromosomes as we age. So this is looking at the loss of the Y chromosome on the left and the X chromosome on the right in populations as we get older. We lose telomere length. We can predict age accurately from a number of metabolic markers. And so our powerhouse in cells, mitochondria, starts to get a lot of changes as we get older as well. So we can put all these together, and we're pretty accurate. It's more or less plus or minus 5 years. But if you'd asked me 15 years ago how many times you'd want to get your genome sequenced, I would have said once. And I never imagined that we would be able to track changes precisely in the genome, and we need to know people's exact age when the blood sample was drawn, because it's that predictive. So we want to see if we could use machine learning to predict something more complex like the face. And so we did a clinical trial of 1,000 people in San Diego where we took 3D photographs, measured a number of things, and sequenced their genomes, and started to put things together. And I will tell you that your genome predicts what you should look like just post-puberty. In my case, that wasn't age 49. I was a late bloomer, but not that age. But they just look for a photo. So this was some of the earliest predictions where they were using just the triangle line, the computer prediction with an earlier photo. Now we predict things from around age 18 or so, and then based on your predicted age from the genome and your predicted BMI, we have an algorithm that ages the face prediction appropriately or inappropriately, depending on things. So you can find out if your diet really goes crazy what you would look like. These are in the earliest stage, but they're getting extremely accurate. And here's some of the earlier studies with some mixed ethnicity populations. The top are the 3D photographs, and the bottom are the straight computer predictions just from her genetic code. Here's another one. So our algorithm right now predicts your face as a symmetrical object. Everybody knows not everybody has a symmetrical face. And the algorithm smooths it. So a lot of people like their face predictions because they're symmetrical and they're smooth. You can see the 3D photo is a little rough, so they look better when they're smooth. But the predictions keep getting more accurate over time. And just straight predictions with no other input other than the ACs and Ts of the genome have been affecting things. So we're compiling all this data on individuals. So each individual, the data files are extremely large. And the things that are enabling us to do the complex computational work on it, for example, if you have a 3 gigabyte brain MRI file, that's not what we're looking at. We take things out of the Neuro Quant that reduces that to a table of exact precision measurements of different brain regions. Every organization has their own matrices, so we have a contest on hippocampal volumes and liver fat. So there's probably an inverse correspondence between the two, but right now, Franz Ock, who's our chief data scientist that I hired out of Google, he built Google Translate. He has the largest hippocampus for a male and the largest hippocampus for a female associated with HLI is my mother-in-law, which is probably pretty good. At least that's what I told her. But we measure all these things and allows us then, just like we did with the face, to do machine learning predictions of what genetic elements determine your hippocampal volume, determine likelihood of Alzheimer's disease, et cetera. And we're just at the earliest stages because we're still just compiling these incredibly complex data sets that have never existed in science and medicine before. along with the genetic code and the microbiome, et cetera. So, as I said, we're just delivering the first reports. The numbers are really quite stunning. We are discovering major medical issues and predicted issues in 30 to 40% of the walking, healthy, wealthy population. These match the statistics I showed you on the death rates, and they match the cancer rates. When you see them in reality with people you know in the clinic, it's really stunning. But these are life-saving discoveries in some places. We haven't had anybody that hasn't been incredibly excited about finding things when they're actionable. I would say out of the 60 or so major things we found, there's only been probably 2 that were too late stage to be actionable. So it's good news if you discover things early enough to do something about it. We present all this data back in a complex avatar, so we take full body 3D photos of people, and we have an iPad that we will give you back with all your integrated data that you can explore around. And with these avatars, we can animate them in any way you want. So if you want your spouse's avatar to do unusual dances or something, we will take those requests for extra money. So we're integrating all this data. The goal is to have at least 1 million records by 2020. I think with our scale up of what's going on with cancer and all these, I think we're going to exceed that. So it's an exciting venture that I hope some of you will have the chance to join. Thank you very much. So we have time for questions. I don't know if we can have the house lights up at all. And I'm supposed to ask you to wait for the roving microphone so that the cameras can hear the questions, not just so that I can. Is it possible to get the house lights up a little bit so I can see? I can see one hand in front, but that's all I can see with the spotlight.
To have the microphone, so. Has your facial recognition program been used in any criminal cases?
We are in discussion with various government agencies and a number of law enforcement groups. Obviously, we can take DNA from a bone sample and generate a picture and a description of that person. I think these things will be very useful at this stage for identification of people. It's not at the level where it's ready to stand up in a court of law compared to the DNA markers that have been tested for a long time. I think they will progress to that level. But yes, I think there's extreme interest in this. So if you imagine from a rape kit or any DNA sample having an actual photograph as well as an pretty exact description of a person and their height, weight, ethnicity, et cetera.
Dr. Ventnor, really amazing presentation. Thank you very much.
Thank you.
For the average consumer, what does it cost for someone to walk into your facility and have this done today?
Well, it costs nothing to have them walk in.
Once they're there.
They might get out with less. It really depends on what we're talking about. So a while ago, we announced our first major insurance deal with Discovery Health in South Africa and England, where they're helping to subsidize it. We're doing whole exome sequencing on their clients for only $250 a person. The Health Nucleus, this complete comprehensive program, costs $25,000 a person. Our comprehensive cancer program, we're charging $25,000 a person. It's probably costing us about $50,000 a person right now, but we'll get much more efficient with that. A whole genome is about somewhere between $2,500 and $5,000. But we're going to have new ways to get exomes and genomes faster and cheaper, and the cancer program will be announcing that in the not too distant future will be much more cost effective. So it's a range right now. Not everybody wants the comprehensive detail that we give in the health nucleus, but if you're 50 or older, provides incredible baseline data that's useful going forward. And we have several insurance companies, so if you have very large insurance policies, your insurer may contact you, and some of them are considering paying the $25,000 to go through the health nucleus, because your life insurance company actually wants you to live a long time. So they have the same goal that you do. So it depends on what you want. There's a variable cost.
That's a really interesting point, but the insurance companies, I mean, certainly for them, you should be incentivizing them to have patients live longer, not have to get to stage 4 cancer. Unfortunately, they don't always have that proactive, preventative mindset. Right. And how do you change that philosophy with the insurance companies, where they're always thinking about the bottom line today rather than worrying about tomorrow?
Well, we're finding life insurance companies are very interested in this notion. As I said, it's the one place in the insurance industry where the insurance company and their client have the same goal, maybe for different reasons. You want to live a long time, they want you to live a long time. I think it's generating this data at the level where it becomes unequivocal that if you do X, it will save you more money than sitting around waiting. It's a difficult thing to do. We're not a species that I think is good intellectually at preventative medicine. We have to really focus on it, because I think there was a recent thing in Congress where I think it's a great example of wanting to eliminate the air marshal system because there haven't been any hijacking, so obviously it's a wasted system. When you do preventative medicine, you don't have disease any longer. People forget that there was a cause of the disease, and that's why there's a problem with vaccination right now. Is there hasn't been any massive outbreaks of polio and things now for multiple decades, so we lose that memory. So it has to be totally financially driven based on clear-cut statistical data is my view, but it's going to be an uphill battle until the data is overwhelmingly out there.
Really great talk. Thank you very much. So with over 7,000 rare diseases in the world, your suggestion of 8,000 mutations that make someone an individual would probably suggest that those 7,000 is a gross underestimation. And with so many X-Men movies and TV series like Heroes, when can we expect to identify more genes that give us superheroes?
I don't know, the whole country seems to be going in the other direction right now. There's certainly some genomes that would be very interesting to look at the current time. And maybe with CRISPRs we can solve some of the problems, but I think we're at the earliest stages, and that's where I go back to where I started this talk with the synthetic cell with just 400-something genes, where we're still learning what a third of the biology does. So I'm one of the few that's written that he was against using CRISPRs to edit the human genome right now, even though it's very tempting to try and, quote, cure a disease by just changing the genetic code. We are a very complex genome-driven species that we really don't understand the genome and how it works. I think there's going to be some very exciting opportunities in the future to edit the genome once we understand it better, and understanding the unique combinations of these rare variants. So, the Delleman syndrome case wasn't a single gene, single disease. It was about 20 genes involved to give a very complex syndrome that you can see mathematically why out of 8 billion people, there's only 30 people with that combination and that disease. When we give out these genome reports, because we're the only place on the planet that can accurately determine allele frequencies, for each thing we find, we report the allele frequency in our database. And if it's a single integer, or less than 50, we take it seriously, and we encourage people to take it seriously. If it's a common variant where the numbers in 19,000 people out of 20,000 people have that same variant, you can be pretty confident that's not the cause of your trait or your disease. Whether we find things that cause super positive effects or longer aging, now that we can predict the age from the genome, we're hoping patterns will emerge that we can at least long-term know how to select things. We have a very unique selection where we think we can, in the near future, tell you how long you're likely to live. And we do this simply by gene traits that we do not find in an older population. So it's not by what people have, it's what you don't find in certain populations. So these large databases are totally changing how we look at all of our data, and I think it's going to be pretty fascinating.
Hi, Dr. Waiter, great presentation. I had a question about gene splicing or slicing for therapeutic purposes. Have you tried anything in your cancer program regarding the gene splicing?
Well, I was referring to that with the CRISPRs that can edit the genome, and it's a great tool working with experimental systems, and it's not hard to change the genome in embryonic cells or some other cells, but as I said, it's very premature to do that when we know less than 1% of the knowledge of the genome. So I think we need to accumulate a little bit more data first. Thank you all very much. Oh, okay.
Talk a little bit about, as we're creating life and extending life, where are the policy issues behind that? Or is that something that you worry just about the science, or do you have any concerns about policy keeping up with the lab?
No, it's a great question, Ted. So it's the policy issues are things we try to very much stay ahead of. We just hired the senior person from the Obama administration that he had sent to the FDA to build the personalized medicine program at the FDA. We worked with the administration when we announced the first synthetic cell, and that arranged for the announcement that he would have his new ethics commission look at synthetic life and the issues around it. It's very key when we go into this stage of increased fundamentalism and people are afraid of science to not have the policy keep up with the science data. And at the same time, something like CRISPR sounds very seductive if you have a disease in your child that you think you can edit a base pair and cure that disease. It's very tempting to at least try to do that, but it's because we don't know the consequences. It's like the metaphor of you want to take new drugs when they first come out because you don't know any of the side effects yet. That was a joke. So that's the safest period to take a new drug because nothing's known. So policy is very key to the future of this, and that's why I think even the government's not doing much in the true personalized precision medicine space other than talking about it. The fact they're talking about it, I think, is good because it raises the discussion.
Okay, thanks.
Thank you.